Okay, guys, we're back. We're with concept 8.3, and it says ATP, which is the adenosine triphosphate molecule that we've talked about in the past. ATP powers cellular work by coupling exergonic, remember that's a reaction that gives off energy, to endergonic, that's a reaction that takes in energy, reactions. So ATP powers the cell by coupling an exergonic reaction to an endergonic reaction. When we talk about cells doing work, we can kind of categorize that in three ways. Some of the work that cells do is chemical related, the building of molecules, like the building of proteins from amino acids, or the building of a polymer from monomers. That's one type of work that cells can do. Another type of work is transport, the motion or the movement of materials against the concentration gradient. Remember, we talked about the cell membrane, and so we're not talking about just diffusion or osmosis here. We're talking about the active transport of a material across the membrane against its concentration gradient. Remember, I said that required energy. The third type is mechanical, and that would be like motion. So the little beating of the cilia of a paramecium to move him around, or the tail of a sperm cell wagging its flagella to move around, the contraction of muscle cells. That's all mechanical work that cells have to do as well. We already know that ATP is kind of like the currency or the cash, the energy cash to get things done in the world of cells. And the way this works is we couple two reactions together. The cells tend to couple a reaction that gives off energy with a reaction that requires the intake of energy to build something. I always try to think of different sorts of models or analogies. So imagine that you have a well that is drilling for oil, and that well uses a motor that's powered by gasoline, or an engine powered by gasoline. So you are using an engine powered by gasoline to drill for oil. That's kind of the coupling that we're talking about. So you are burning fuel to get fuel. You are expending energy to store and create energy. And again, when I say create, I don't mean make it out of nothing. I just mean store it, transform it into a mode that you can use. The molecule that is at the heart of all of this is the ATP molecule, the adenosine triphosphate molecule. That's the cell's energy shuttle. It's the one that is involved in all of this process. We already know that it's composed of a ribose sugar, an adenine, which is a nitrogenous base, one of the nitrogenous bases we looked at, and three phosphate groups. Here's a quick model right here. So this is ATP. Here is the ribose sugar. It's got the circular carbon molecule shape there. Here is the nitrogenous base, the adenine, and here are the three phosphate groups stuck onto the end. These three phosphate groups, particularly the last group here, is probably the most important part, because this is where the energy is released or placed back into the molecule itself. So let's look at the hydrolysis of ATP. Remember, hydrolysis is adding water to break the bond. So here we have the ATP molecule. We add water. That's going to break off this phosphate, this third phosphate right out here, that releases energy. So the hydrolysis of ATP releases energy, and that's the energy that can be used for the cell to do its work. These products at the end of the reaction have lower free energy than the ingredient that we started with. So these products end up with lower free energy. So where did that energy go? Well, it's here, and it can be used for a variety of purposes. So the bond between those phosphate groups, that last phosphate group, can be broken off with hydrolysis, and that releases energy that can then be used for a wide variety of cell work. So in the cell, the hydrolysis of ATP, which is the breaking off of that last phosphorus portion, that's by adding water, that releases a large amount of energy. And then that energy can be used to do something else. That energy can drive an endergonic reaction, a reaction that requires the input of energy. Now, if you coupled these two together, we would still say that overall it's exergonic. In other words, you are giving off more energy than you're taking in. So overall, this coupled reaction is still exergonic. So again, I try to think of silly examples when I can, or silly ways to think about it. So let's say that you take a donkey up to the third story of a building, and you tie a rope around him, and there's a rope and a pulley. And on the bottom floor, on the street level, you have your little dog. Let's say it's a poodle. And your job is to lift that poodle to the second floor. So you take that donkey and you toss that donkey, tied with a rope of course, out the window on the pulley. As the donkey falls, he lifts your poodle up to the second floor. So you kind of have a coupled reaction. You have the donkey falling from the third floor down to the ground. You have the poodle rising from the ground up to the second story. Now, overall, did you lose or gain? Well, overall, you would say you lost. I mean, the donkey went all the way down 3 floors. The poodle went up 2 floors. So overall, you lost. But you did get something accomplished because you did get your poodle moved up to the second story. So the donkey falling down 3 floors is kind of like the exergonic portion, and the poodle moving up 2 floors is the endergonic portion. But overall, if you looked at the entire system, you still lost energy. So how does ATP drive an endergonic reaction? Well, we said through hydrolysis, one of those phosphorus or phosphate groups are broken off. That phosphate group can then bind with another molecule, and that's called phosphorylation. And when it binds with that other molecule, it sets it up to go ahead and go through its own reaction. So when that phosphate group is broken off at the end of ATP and is releasing energy, that phosphate group will then bind with the recipient molecule, and now we call that a phosphorylated intermediate. And doing that makes that intermediate molecule more reactive, less stable, so then it is more likely to go through its reaction that it needs to go through as well. Let me show you a couple of examples that we've already talked about. So remember we talked about these transport proteins. The transport proteins change shape, and in this case this would be active transport. We're going against the gradient. So see, we're moving these little brown dots outside of the cell where there's already a higher concentration. So that's going to require the input of energy. And so the ATP molecule will be hydrolyzed. It will have one of those N groups broken off, the phosphate N group broken off. That phosphate N group will attach to the protein. That will change its shape and allow it to move that solute from one place to another. And then that phosphate group is then released from the protein. Note that at the end of this, we have ADP, adenosine diphosphate, and we have that extra phosphate group. Those two can actually be recombined later to create ATP and start the process all over again. Here's another example down here about the motor proteins that kind of walk along the cytoskeletal tract we were talking about. That's going to require energy. So we have a little motor protein right here, ATP, that is going to be hydrolyzed. That phosphate group will be attached to the protein, and that will cause it to change shape, which will cause it to walk along this cytoskeletal tract. And then after that occurs, we have ADP, adenosine diphosphate, plus that phosphate group that was taken off. This can be later recombined to create ATP and start the whole process over again. ATP is a pretty cool molecule. You can kind of think of it as a renewable resource because it can be regenerated and reused. When it gets hydrolyzed and that one phosphate group is broken off, it becomes ADP, adenosine diphosphate. But that ADP can be phosphorylated itself. In other words, we can have that phosphorus group added back to it, and it can become ATP again and start the whole process. So the energy that it takes to recreate the ATP comes from a catabolic reaction in the cell. It's kind of like recharging a battery for another use. So if you have rechargeable batteries, you have to understand they're not 100% efficient. So the electricity that you get from the wall to recharge those batteries, the battery will not be able to use and store all that electricity that comes from the wall. So overall, you're still losing some energy, using the energy, but you can recharge and reuse that battery over and over and over again. So ATP is kind of like a revolving door, which energy is transferred from catabolic, which is the breaking down and releasing energy, to anabolic, which is the taking in energy, building up pathways. So here we go. We have the ATP, kind of like the Lion King circle of life here. So this is energy from catabolism, which is the breakdown. So let's say you're digesting food, and those digested food molecules can be used to create energy or to release energy. So here we have ATP. That ATP can be hydrolyzed. That energy can be used to build. So your body can build new skin cells, for example. And then that ADP can be phosphorylated again, and the whole process can start over, round and round it goes. The last portion of this chapter is all about enzymes. Enzymes are proteins. They are proteins that are what we call catalysts. Catalyst, remember, is some sort of chemical agent, some sort of material that speeds up a chemical reaction but is not changed or consumed by that chemical reaction. So an enzyme is the most important catalyst of the human body, or most animals, and an enzyme is a protein that speeds up a chemical reaction. So an enzyme is a protein catalyst. So a good example might be the hydrolysis of sucrose by the enzyme sucrase. You'll notice that most enzymes end with the ASE kind of appended to the end there. That's an example of an enzyme catalyzed reaction. If you were to put sucrose, which is table sugar, into a glass of water, it will break down over a very long period of time into more simple compounds. It will eventually break down into glucose and fructose, which are more simple than the sucrose is. Remember, the sucrose is a disaccharide. So this can happen automatically. This is increasing entropy. This would be a spontaneous reaction. But it is very, very slow. Way too slow for this to be relied upon in the human body. Now, if you were to take that same cup of sucrose, sugar water, and you added an enzyme called sucrase, suddenly this speeds up the process tremendously. So it's not changing the process, but it is speeding up the process quite a bit. And the way it works is, we'll find out, enzymes have the ability to basically bring things together, or in this case, break things apart and speed up that process. So why are enzymes so important? Our body relies on a huge number of enzymes to catalyze reactions. And why? Why can't we just let things proceed naturally as they normally would? Well, the problem is there is this, what we call this activation energy barrier. Think about a roller coaster, how a roller coaster coasts, the old style roller coaster, like the wooden roller coaster. It coasts the entire way, except for the fact that it has to get over that first hill. And to get to the top of that first hill, it has to use a motor or a pulley or a chain or something to get it up to that top of that first hill. Now, once it gets up there, it can go the rest of the way on its own. That's similar to a chemical reaction. This first hill that it has to get over, this hump that it has to get it started, is called the energy barrier, the activation energy barrier. So every chemical reaction, even chemical reactions that will occur spontaneously, have to overcome this initial activation energy barrier just to get the whole process started. Usually they get that energy from their surroundings, just from the thermal energy, the random motion of the molecules and atoms in their surroundings. But sometimes that thermal energy of their surroundings is not high enough to overcome this first initial barrier, this first initial hill. So let's say you have an exergonic reaction. You have these particular reactants that eventually will combine to create these products. Now this is an exergonic reaction. In the end, we have a change in free energy that is negative. So this should proceed naturally, spontaneously, all by itself. But there is this transition state, kind of this portion where we have to get things going. That requires a little bit of an input of energy. Now once it starts, it'll proceed all by itself, but we have to overcome this need, this activation energy first. What the enzyme does is it lowers that first hill. It doesn't change the process, but it lowers the energy of activation barrier. So it makes that first big roller coaster hill into just a little speed bump, and the rest of the roller coaster gets to proceed as it usually would. The enzyme doesn't affect the change in free energy. All it does is lower that first initial hill to speed up the reaction that would have occurred eventually on its own if there was enough energy, say thermal energy, in the environment. Here's a quick little video, a little animation. You can hit right click and play, and you can see how what the catalyst does, what the protein catalyst, the enzyme does, is it lowers the activation energy. And one way to think about this, again, going back to the analogies that I obviously love, consider an enzyme kind of like a matchmaker. So perhaps you and your girlfriend or boyfriend or your significant other, maybe you would have met on your own, maybe you would have fallen in love all on your own without anyone's help. But the whole process of you meeting your significant other, perhaps that whole process was made easier and sped up by this mutual friend that you both had. And that mutual friend introduced you and kind of set you up and sent you out on a first blind date and talked to you both and encouraged you both to get together. Well, that matchmaker is kind of like this enzyme that we're talking about. The matchmaker didn't change what probably would have happened anyway, perhaps, but it sure sped the process along, and it lowered that initial hill that had to be gotten over for this process to continue. So the matchmaker, him or herself, they're not changed. So it doesn't, if you were to get together and eventually get married, fall in love, etc., the friend is unchanged in this situation, and the friend is free to act as a matchmaker with another potential couple. So if this friend likes matchmaking, they can go and create couple after couple after couple. That doesn't change the friend. So just like the enzyme isn't changed, and the enzyme is free to cause this reaction to occur over and over and over again, that's kind of what we're talking about. Oh yeah, I forgot about the animation. So hit right click and play. So here we go, just another graph. I know some people like to see the graphs. So here we have a reaction. Here's how it would naturally occur in black. This would be the natural occurrence without an enzyme. And then here, the red is with the enzyme. Notice the reactants start out with a certain amount of free energy, and they have a negative change in free energy. They end up at the same spot. So it doesn't change the reaction. All it did was lower this initial energy of activation that was required to get it started. Some vocabulary words that we kind of need to know. The reactant, or the material that the enzyme acts on, is called the substrate. So in our matchmaker analogy, you and your significant other would be the substrate that the enzyme acted on, and the enzyme is the matchmaker. The enzyme usually will bind to the substrate and form something called the enzyme-substrate complex. This would be like if your matchmaker friend suggests that you and this person who becomes your significant other, you all go out to the movie together. So you go to the movies. It's kind of a semi-blind date, but your friend goes with you so you don't feel uncomfortable. So your friend sets you up with this other person. You all three go to see a movie. Well, that would be like the enzyme-substrate complex. The spot on the enzyme where the substrate binds is called the active site. Ooh, don't know if I can come up with a good analogy with this one. Let's say the movie theater itself is the active site. So the movie theater isn't part of your matchmaker friend, of course, but we'll pretend that it is. So that's going to be considered the active site, the region of the enzyme where the substrate binds, the place where the matchmaker got the two of you together. And finally, there's this word called induced fit. Induced fit is basically when the substrate is brought into a particular position that helps speed up the reaction. So in a moment, we're going to talk about protein shape. And remember, proteins have all these complex shapes, and enzymes are proteins. So the shape has a lot to do with it. So one of the reasons the enzyme speeds up the reaction is because it brings the substrate molecules together in a certain way so that they are fitted together, and it speeds that process along. So let's say when you get to the movies, initially you sit on the right and the blind date person sits on the left, and the matchmaker friend of yours sits in the middle. Well, your matchmaker friend may say, oh, can you scoot over? I'd like to sit on the outside. And so your matchmaker friend scoots over and forces you and the blind date person to sit next to each other. That would be like induced fit. She is putting you together in a way that will encourage this reaction. Here's a model showing an enzyme. This giant, big, massive blue protein is the enzyme. The active site where the substrate is going to bind is right there. Here's our little substrate. And notice what happens when the substrate binds. The enzyme sometimes changes shape slightly. And here we have the enzyme substrate complex, and it brings that substrate material in closer contact or in the right shape or position for the chemical reaction to occur. So how does that enzyme lower the energy of activation barrier? Well, there's a couple different ways. First, it can orientate the substrate. In other words, put it into the proper position so that it makes it easier for the chemical reaction to occur. The second one is by straining the substrate bonds. The protein will actually kind of bend or twist or move the substrate so that it basically kind of physically starts the breaking process for that chemical reaction to occur. The third is by providing a favorable microenvironment at that activation site within the enzyme. There may be a particular set of environmental factors like pH or something to that effect that makes it more likely that the process will continue. And then finally, sometimes there is actual covalent bonding to the substrate. So sometimes there's an actual chemical bonding that occurs temporarily between the enzyme and the substrate that will help speed the process along. So here we go. Here we have a cool little model. Here's our enzyme. This would be a protein. It's got an active site. The physical shape of it does matter. Here we have our substrates. These are the two things that we want to get combined. They will bind in here to create an enzyme substrate complex. Notice that they are not combined yet chemically, but they are held together by some weak interactions, hydrogen bonds, van der Waals interactions, things like that. Next step is that because of this protein in the active site, it lowers the energy of activation enough that the reaction can occur. So now the reaction has occurred, and the substrates are converted into the product. They are released, and the enzyme is free to do that process all over again. Enzymes can be affected by the environment as well, so factors such as temperature and pH, chemicals, things like that can influence the enzyme, can make it either more active or less active, can shut it down or let it work better. Temperature is a big one because we know that particularly high temperatures, remember enzymes are proteins, and high temperatures can do what we call denature proteins, which causes them to unfold and lose their proper shape and orientation, and that would certainly ruin the whole enzyme effect. Every enzyme has a particular temperature that it works best at, an optimal temperature. Enzymes have a particular pH that they work best at, and if you want this enzyme to work as efficiently as possible, there's going to have to be optimal conditions for that particular enzyme. You can probably guess. I mean, if I asked you at what temperature do you think that most enzymes found in the human body work best, well, obviously, for most of them, it would be right at about 98.6 degrees Fahrenheit. One of the problems with getting sick and running a high fever is that inside your body, once your fever begins to rise to 101, 102, 103, that can begin to denature some of these proteins, denature some of your enzymes, and that starts all sorts of problems where your enzymes are not properly working to allow the chemical reactions necessary to keep you healthy and alive. Different enzymes in different creatures and even in different parts of the body of a different creature have different environmental requirements. So like the optimal temperature for most human enzymes, you'll notice is right about 37 degrees Celsius, which is about human body temperature, whereas the optimal temperature for the enzymes of thermophilic bacteria that lives in hot water springs and things like that is closer to 77 degrees Celsius. 77 degrees Celsius is about 170 degrees Fahrenheit, which is amazingly hot. It's kind of interesting to think that there's bacteria that live in these hot water pools, geysers and things of that nature. So you can see where if this bacteria that lives in a hot water pool of Yosemite National Park or something like that, if they had human enzymes, that would be a big problem because they live in water that is quite a bit hotter than 98.6 degrees. And so those enzymes would denature and would be useless to them. pH also, this can occur inside even your own body, in different parts of the body. Inside the stomach, pepsin, which is a particular stomach enzyme, works best at a pH close to 2, which is very acidic. Then if we go further down into the intestinal tract, into the small and large intestine, there's another enzyme called trypsin. it actually works best at about pH of 8, which is more basic. Enzymes sometimes have helpers. So again, going back to our matchmaker scenario, let's say your matchmaker friend has a little sister that is helping her set up this whole blind date. So her little sister would be called a cofactor. So if an enzyme has a helper, a non-protein helper, it's called a cofactor. The cofactor can be inorganic. In other words, it's not carbon-based, so it can be a metal, an ion, something like that, or it can be organic. If it's organic, it's called a coenzyme. So cofactors can be inorganic or organic. If they are organic in nature, they're called coenzymes. Vitamins in the human body or in animal bodies, vitamins are a good example of coenzymes. A lot of the vitamins and nutrients that you get from fruits and vegetables and things like that are coenzymes. They help the enzyme achieve its purpose or do its job. There are other materials that work against the enzyme. These would be enzyme inhibitors. And there's a couple ways that can happen too. You can have what's called competitive inhibition or competitive inhibitors. What they do is they bind to the active site of the enzyme and take up that space. So if this competitive inhibitor is already in that space, in that spot, well then the material that normally goes in there, the substrate, is not going to be able to get in there. Another example of this, or type of this, is called non-competitive inhibitors. And this would be where a material binds not to the active site of the protein, not to the active site of the enzyme, but another part of the enzyme. But binding to that other part causes the shape of the enzyme to change, and then the active site is no longer the right shape or size for the substrate to get in there and create the reaction, making it less effective, basically. Some good examples of inhibitors, competitive inhibitors or non-competitive inhibitors, all sorts of toxins and poisons and pesticides and antibiotics. These are all enzyme inhibitors. And I don't want you to think that all enzyme inhibitors are these deadly materials, like poisons, because your body also produces and uses enzyme inhibitors itself to slow down the speed of reactions. But oftentimes these enzyme inhibitors or bad things that come from the outside. A good example of a non-competitive inhibitor is sarin gas. Maybe in the news you've been watching about what's going on in Syria lately, and the claim that the Syrian government has used poisonous gas, including sarin gas. Sarin gas is a non-competitive inhibitor. It attaches to a particular enzyme, not at the active site, but at a different spot, but it changes the shape of the enzyme so the active site doesn't work very well. And what this does is it blocks the working of an enzyme that's very important in the nervous system. So it leads to a fairly quick death. Here we have a couple of models. Here we have our enzyme, and this would be our substrate, and this is the active site. This is normally what happens. That's good. That's what we want to see. Here we have two types of inhibition. Competitive inhibition is where some inhibitor has gotten in the way and kind of blocked the active site, so the substrate can't get in there. Non-competitive inhibition means that the inhibitor has attached somewhere else, not at the active site, but somewhere else, and that has changed the shape or the location of the active site. See how it's not exactly the same as it was? And now that makes it so that it's not as good for completing that reaction. Either the substrate can no longer bind there, or it can, but it doesn't happen as often or as quickly. How did these enzymes come to be? How did our body create these enzymes? Or how did living creatures, not just us, create these enzymes? Well, remember, enzymes are proteins, and proteins are encoded by our genes. Our genetic material tells our cells what proteins to create, and enzymes are a type of protein. So via evolution and genetic changes, sometimes you have what's called mutations, and a mutation can lead to a change in the amino acid chain that makes up a particular protein. So you may have a protein that is created what you could consider mutated or abnormal, and if that abnormal created protein turns out to have some function that is useful in a particular environment, well then it may, over time, become more common within that species. So while we generally think of the word mutation as being bad or negative, and sometimes it is, some mutations can be beneficial. So if there is a coding problem and a protein gets misshapen or created in some new and novel way, and perhaps it's going to be random chance, but in some cases that new protein, that new enzyme, has some sort of favorable effect, well, that's good. That's going to help that particular creature or that particular cell. And that may be then passed on to the offspring. Over time, you may find that that particular protein or enzyme becomes more and more common. That is the process of evolution, change over time. The very last part of Chapter 8 is about the regulation of enzyme activity. So your body has to have a way to stop and start, slow down, and speed up this whole process. Otherwise, we would just have chemical reactions gone crazy. I mean, our whole body would just be chemical reactions occurring at lightning speed, and it would be very difficult to control all of these processes. So the body does have ways of switching enzymes on or off, or slowing them down, speeding them up, regulating the enzymes. One of the ways that your body or living creatures, I keep saying your body or human bodies, but this applies to all living creatures. One of the ways that the enzymes are regulated is called allosteric regulation. Allosteric can be positive or negative. In other words, it can inhibit the protein or it can stimulate the enzyme protein to do its job. So it's just a form of regulation, but it can be a form of regulation that moves the process along and speeds it up, or it can be something that slows down the particular enzyme's activity. So allosteric regulation is where some type of molecule, a regulatory molecule, binds to the protein at one site, and that affects another site of the protein. So this would be similar to that non-competitive inhibition we were talking about, where the inhibitor binds to not the active site, but a different site on the protein, and that changes the active site. Well, this is similar, but when we say allosteric regulation, we simply mean binding to one part of the protein, changing the active site, either in a good way or a bad way. Most of your enzymes that are allosterically regulated are made up of these long polypeptide subunits. And each enzyme has a couple different forms, has an active form and an inactive form. And the binding of the activator will stabilize the active form. In other words, it will put it into a positive position where it can do its job well. And the binding of the inhibitor stabilizes the inactive form, and otherwise it puts it into a position or shape where it doesn't work at all or doesn't work very well. So here's a good example. So here we have two enzymes, and they have two different possible shapes. Here's the active form up here, and here is the inactive form. And they can take either one of these shapes. So here we have a particular activator. This little activator binds at this spot, and it kind of locks this enzyme into the favorable active form. So then it can do its job at all these different little active sites. The other form, the inactive form, if this inhibitor binds, it will stabilize the inactive form. And now notice all those little active sites are not the proper shape to allow for the reaction to occur. So this is allosteric regulation. Allosteric inhibition down here, and allosteric activation up here at the top. One last form of allosteric regulation to talk about is called cooperativity. Cooperativity is basically where you have a substrate molecule that will bind to the enzyme at one of the active sites, and that further enhances that enzyme and its ability to do its job. So cooperativity is a positive allosteric regulation. It is enhancing the active sites of the enzyme. So we may start out with an inactive form of the enzyme. This substrate is able to bind to one particular spot, and that then creates all the rest of the enzyme, opening these active regions and allowing for the enzymatic action to occur. So the reason we still call this allosteric is, yes, the substrate here is binding to an active site, but then this binding here is affecting all the active sites at different locations. So allosteric means you're binding in one spot and affecting another spot. Usually we talk about binding to a non-active site and affecting the active sites, but here we're binding to one active site and affecting all the other active sites. These allosteric regulators are very attractive for drug candidates, for enzyme regulation for drug companies and pharmaceutical companies because they're very, very specific. Remember, I mentioned that these enzymes are very specific as to what particular substrate they'll bind to and help along with the chemical reaction. So if you can develop a drug that perhaps will modify an enzyme or change an enzyme and either activate it further or deactivate it, you can have a very specific effect on cells or the body, and that's good. If you're taking a particular medicine or a drug, you would like it to be as specific as possible. And so these allosteric regulators would possibly make for very good drugs if we can control that and regulate exactly which enzymes get turned on and get turned off, because that would help with particular symptoms and various other health problems. Some of these allosteric regulators or some of these different ways of basically shutting down enzymes or slowing down enzymes, they're not all negative things that come from the outside. We talked about how the human body, again, I keep saying human body, but all living creatures, they have to have some way to regulate this whole mechanism. So sometimes they use negative feedback, which we already discussed, and that's called feedback inhibition. So feedback inhibition is where the enzyme process gets slowed down or shut down by the very material that it's helping to create. So feedback basically keeps the cell from wasting resources, from wasting energy and chemical resources by making more of a product than it needs. So here's a quick chart that kind of describes what we're talking about. So here we have the initial substrate and the enzyme that we start with, and this is where the initial substrate binds to the enzyme at that active site. And then there's a bunch of steps along the way, but the final product is this particular molecule. In this case, it's called isoleucine. This isoleucine begins to build up in the cell environment. So the enzyme creates more and more, and it's going through all these steps, and more this isoleucine builds up, builds up, builds up. Well, eventually you get a large enough amount of this material that it starts to bump into these enzymes themselves, and it binds to a different site, an allosteric site. And when it does, it causes a change in the active site. So then notice how the shape is changed. When that occurs, this substrate can no longer attach, and so it shuts down this whole process. So basically the cell is going to create this product, create, until there's so much of it that it starts to interfere, and it kind of shuts the whole process down. Well then, as the body uses up that product and there's less of it, well, you'll have less of it binding to the enzymes, and suddenly that enzyme is available to use again. So the body has a way of slowing or stopping this process until that product is used up, and then it can start the process all over again. This is a great example of a negative feedback inhibition. The last and final thing we're going to say is that sometimes the way that the cells or the organism will help control this whole process is by keeping enzymes in certain areas or within certain cells or certain cell structures. That way these enzymes are not free just to speed up these chemical reactions everywhere. You kind of have a controlled process to where they're kept in a certain spot. So oftentimes, like in eukaryotic cells that have all the different organelles, you'll find specific enzymes in specific organelles. So enzymes for cellular respiration are located in the mitochondria, while enzymes with a different purpose are located in a different organelle. So these enzymes are not just everywhere. They're generally located in specific parts of the cell or specific parts of the body having to do with their function. And then one last little picture here. So here we have a model and then a microscope picture of a mitochondria. And they talk about how you have the two parts of the mitochondria. You have that inner space, and then you have the folding of the inner membrane. And there's actually two different enzymes located in these two different areas. And the two different enzymes, of course, have a different purpose. And that's why they're kind of segregated into their separate areas. So that's another way that the body can help regulate this whole process. All right, that wraps up Chapter 8. I know this was I kind of rushed through this very fast. I'm sorry for getting this up a little late, but a lot of this we're going to kind of cover as we continue to talk about these processes. The processes coming up, we're going to talk about cellular respiration and photosynthesis, and we'll mention a lot of this material again as we go. But it's helpful to kind of do a quick review, particularly about the importance of these enzymes, these proteins that help speed up these reactions. Pretty important to understand that process. All right, see you guys in Chapter 9. Have a great weekend. Talk to you later. Bye-bye.